end of 2024.
Okay. Good luck.
Thanks.
Yeah, so I thought maybe as a start, because we thought about it, and in biology, because I graduated from this building. I got my PhD from here, doing statistics with numeric methodology. And now I'm moving to biology, computational biology. And one thing I noticed in biology that first people don't call their groups labs. You could have a real, like a wet lab, or it's just a dry lab with computers, but they call it lab. And we have regular lab meetings, and on that kind of meeting, we can do all sorts of things. Can be a presentation, can be a discussion. So I thought maybe for ECAs, we could have something similar. So for ECAs from different background, we do some talks, and talks can be very flexible. I mean, I aim at 30 minutes, but it could be longer, could be shorter. Anyway, it's more like a networking event. So I'm thinking about having it monthly, roughly, or more, or we can insert or delete sessions depending on, yeah.
I quickly turn off the lights. I'm finding it difficult.
Oh, sure. No problem. Yeah. Yeah, so everything is flexible, and the most important thing is for people to know each other, know each other's face, know each other's work, and maybe know each other's building, because we can have it in different buildings. For example, Emilia, you said you from old geology. Yeah, we can have. Do we have like seminar rooms like this?
Yeah, there's a meeting room. We don't have the best facilities, so but
yeah. So yeah, and maybe some collaboration, because one thing I noticed, because when I was a PhD student here, I actually didn't know I knew the existence of MIG, but I didn't know anyone from there, and I didn't know what they were doing. So, but actually there is a lot of opportunity for collaboration, because basically the problem is in biology, we have a lot of data, all sorts of data, and people use all sorts of tools from applied math and statistics to analyze them. So yeah, if you simply want to find an area of application, then maybe you could consider MIG to work on genomics. And welcome to join a Slack channel. So I will share the recording and slides, etc., in the channel. Yeah, so one thing I noticed this week, because I attended the school, I mean the lunch at Prince Albert Hotel last Friday, organized by Daniel. Did you guys receive that? No?
no.
Oh, yeah, I might invite you guys to because we have another Slack channel for stats people, stats and data science people.
I don't really do any stats.
Oh, so that's why.
Yeah.
You're from applied math?
Applied maths, yeah.
Oh, okay. Oh, yeah, that's for stats people. Yeah, anyway, so I attended that, and I talked to someone, and I was trying to explain what do I do. And I said I do computational biology, but what does that mean? And he said, is it mathematical biology? And I said, well, a bit different, because at least for me, mathematical biology, if I need to think about keywords for mathematical biology, it's probably differential equations and disease modeling, epidemiology. So like the sort of thing James Markell does, for example. That's just my impression. And if you talk about biostatistics, it's more like in the school of public health. People do clinical trials, experimental design, and they do population genetics, meaning you look at a disease, for example, breast cancer. You look at patients and the family history of breast cancer, etc. So you look at individual patients and their relatives. And bioinformatics is more like, so people use, this is more like data preparation. So we have, for example, in RNA sequencing, DNA sequencing, we have a machine, and biologists prepare samples and to be sequenced. And then from the sequence, the output is not a nice data matrix. The output is actually quite messy. So that's the job for bioinformaticians. So they convert the results from the machine to a nice data matrix, M by P data matrix for statisticians to use. And they're quite important because their work determines the quality of data. So it's more like a data preparation step. And computational biologists. So most computational biologists that I met work on things related to cell and at a cellular or molecular level. So the key words there could be sequencing, DNA, RNA, proteins, etc. So yeah, because I just joined our lab 4 months ago, and before that I didn't know anything about these, but now I realize there is a difference between these different names. So yeah, I do computational biology, so it's more like think about sequencing, DNA, RNA, proteins, etc. And yeah, probably skip this slide. And yeah, genomics and statistics. So there is actually, we talk about computational biology. Computational biology has always been an important force to drive methodological research in statistics, in theoretical statistics. Because if you look at, I remember around 15, 20 years ago when I was still in, well, I was in high school, but it was around that time, a lot of people started to do methodological research on high-dimensional statistics. For example, Lasso was proposed in 1996. And that is because in biology, in computational biology, we have a lot of high-dimensional data. And those genomics data are naturally high-dimensional because every one of us has around 20,000 genes. So if you sequence those genes and you get a gene expression level for each of those genes, then for each individual, each person, each patient, you have a long vector of length 20,000. And normally you don't get many patients, you don't get many individuals. So in a study, typically you just get 20 or 50. So your N would be, so if you look at the data matrix, because in statistics, we talk about the N times P data matrix. N is the sample size, P is the number of variables. And if you think about that a matrix like that, then N is typically just 20, 50, but P is 20,000. So this is so this brings a lot of problems. First, if you want to calculate the covariance matrix, the covariance matrix would be P times P, so 20,000 times 20,000. But when you only have 20 individuals, N equals 20, that matrix is singular, so you cannot invert it. You cannot compute the inverse, matrix inverse. But computing matrix inverse is important in statistics. So yeah, and also the data is naturally high dimensional, and it also contains a lot of technical noises. First, so earlier technologies tend to have dropouts, meaning that the gene is actually expressed, but you simply didn't detect it. So you would observe, you would see a lot of zeros in your matrix, because those could be dropouts. And another big topic in computational biology is how to deal with batch effects. So batch effects simply means it can mean many things. So if you look at two data sets, so one data set is like gene expression for one cohort of patients. The other matrix is the same thing, same variables, but for a different cohort of patients. Then can I simply stack these matrix together? So if I have 20 patients in the first study, 20 patients in the second study, can I simply combine them so that I have 40 patients? Sometimes no, because you could be they could be, I mean, bioinformaticians. That's they could so those two data matrices could be from different technologies, and different technologies have different sensitivity and stuff. So basically, if you see, say, if you found, say, 20 copies of RNAs for a gene in dataset 1, and you find 20 for the same gene in the second dataset, you also see 20, but those two numbers might have different meanings. So the equality of numbers doesn't mean the equality of expression due to technical noises. And it also depends on biologists. So even if you are using the same machine, same kind of technology, but different preparation of human tissues could lead to different results. So yeah, so how to combine different batches or different blocks of data to and to get rid of to correct the batch effects. That's another big topic in here. And in particular, I found a paper here. This is from around 15 years ago. It was talking about summarizing statistical challenges of high-dimensional data. And you can see that here it says high-dimensional data can arise in different applications, but the greatest emphasis in terms of applications is on aspects of biology, because it's really because the development in genomics suddenly made a lot of high-dimensional data available, and biologists didn't know how to analyze those data. And also it motivated the development of multiple testing. Multiple testing means you are doing statistical testing, but for a lot of things simultaneously. And here it mentions that for 20,000 genes, also mentioned that number. That's because the total number of genes in each one of us is around 20,000. So a problem with testing in that case is, so why do biologists care about testing? Because they want to know, for example, if we have healthy patients as control and we have COVID patients, and we want to look at all those genes and to see which genes are differentially expressed. So if we see a gene is highly expressed among COVID patients or lowly expressed among COVID patients, then biologists want to look further into those genes. But first, we need to identify those genes. And one way to do that is to do a t-test. So you simply do some transform and assume your data is normal, and you compare for a particular gene, you can do a testing, compare the two groups if the mean is the same, if the means are the same. But the problem is when you do 20,000 tests simultaneously, you're going to have a lot of false discoveries, which means the test says there is a significant difference, but actually that's due to pure chance. So that's false positive. So you have to use some technologies to control to modify your p-values, etc. So yeah, so those two are really are still high-dimensional statistics, multiple testing, are still important topics in statistical methodologies today. And now we have new challenges because biotechnologies are developing very, very fast, and it's faster than statistics, basically. So now instead of large, that's a typo, it should be small and large p, of course. So we used to have a lot of small and large p data, but now in addition to that, we also have big data. So the p remains large because we still have that many genes, but n becomes n exploding because n used to be number of individuals most of the time. Now with single cell technology, n can be the total number of cells. So so single cell technology simply means instead of looking at gene expression, so what you I will explain that maybe earlier. So one analogy we always use is the difference between smoothie and fruit salad. So previously, when I said like 15 years ago, 20 years ago, we had higher dimensional data. We're looking at smoothies. So we take a tissue, for example, if this patient has lung cancer, unfortunately, then we take a piece of that person's lung tissue, and we make it into a sort of biological smoothie, and we sequence the expression levels of all those 20,000 genes. So for each patient, you have or at least for each tissue, you have a vector of 20,000 dimensions. But now with single cell technologies, we can actually separate those cells. So we can look at individual cells and we do that sequencing. We do the same sequencing. So now for each cell, we have a vector of length 20,000. And typically in a single cell study, we are talking about thousands and to millions of cells. So suddenly your N becomes very, very large as well. So nowadays, if you download a single cell data matrix from a website, it's typically gigabytes, 5 gigabytes, 10 gigabytes, huge matrices. And so the matrix you get is like this. I just want to make it absolutely clear. So in single cell, for example, in single cell RNA sequencing, you get a data matrix like this, and somehow they use a different convention. So they put genes as rows and the cells as columns. But in statistics, we typically would do a transpose because we are used to having like observations as rows and variables as columns. But anyway, that's just a matter of convention. So you have genes on 20,000 genes, and you have millions of cells. So you have a huge matrix. But still, I should remind you that the number of individuals is still small. It's just that from each individual, we can look at millions of cells individually. So now from small and large P problem, now we have a big data problem. So yeah, so what do we want to know from those data? First, one of the, so in addition to the testing problem, I said, because we want to find biomarkers, we want to find so-called marker genes by doing those testing. So which genes are significantly differentially expressed in two cohorts across different cohorts of patients. And the other thing is now since we can look at biology at the cellular level, look at individual cells, we want to figure out what kind of cell is this. So if we look at an individual cell, so now we can begin to talk about cell types. So instead of just one patient, one tissue, one vector, now we can talk about cell types. And in immunology, for example, that's very important because we want to first at least classify or cluster the cells, millions of cells at hand, to different clusters. Those could be T cells, B cells, and all those immune cells. And we can even look at the so-called cell states, because not every cell is behaving, has a typical behavior, because you could have, like the T cell for from a COVID patient may be different from a T cell from a healthy patient. So even within those cell types, you have a huge heterogeneity. So we also want to, so instead of just giving a label to that cell saying this is T cell, we want to know what kind of a T cell is that? How do we characterize that T cell? So instead of cell types like discrete labels, discrete identities, now we have a continuum of identities. So we want to locate a cell on that continuum. What kind of T cell is this? This may be a T cell behaving like a, for example, B cell, etc. So In statistics, in machine learning, this is sometimes called a soft classification. So instead of saying this data point is from class 1 or class 2, we say 50% chances from 1, 20% chances from 2, and maybe 20% chances from 3. So we characterize it as a combination of all those known classes. And yeah. And okay, so I've explained single cell, so simply means we look at a range of variables like RNA or proteins. It could be other things, but at a single cell level, at a cellular level. And we could talk about cell types or cell states. And there is another key word in my title, that is multi-omics. So what does it mean? To understand this, first we have to know what I use the factory analogy, because a cell is really like a factory constantly producing proteins. Because proteins are like if you burn your hair, the smell you smell is basically burnt protein, the smell of burnt protein. So proteins are the constructing blocks of our body, and it's also proteins are also tools for cells to do things. They could use proteins as enzymes, for example, to make chemical reaction faster or slower. So they are building blocks, they are tools, and they are like you can think of them as proteins as the products of this factory, and each cell is a factory. And in the formation of, in making proteins, there is a production line. The beginning point is DNA. Every information about the proteins are written in our DNA. And so what about omics? One omics is like a snapshot of this production line, because it's not 2 steps from DNA directly to protein, but it's like a production line. We have many intermediate steps. For example, DNA has to be first to be transcripted to mRNA, messenger RNA, and messenger RNA has to be translated to amino acids, and amino acids need to form a chain and then be folded, because protein, for example, this is the crown on coronavirus, the spikes on COVID-19 on SARS-CoV-2, and it has a 3D structure. But but this 3D structure is actually formed by a very long chain of amino acids. So from DNA to mRNA, from mRNA to a chain of amino acids, and from a chain of amino acids, you experience some kind of folding to have a 3D structure. And I actually omitted a lot of other steps, but you can imagine if we look at every step of this and we measure something, that is called an omics. So the most common kind of omics is probably transcriptomics. In that case, we measure mRNAs. So we look for mRNAs in the cell, and we count how many, because every mRNA, every piece of mRNA corresponds to a gene in the human genome. So we simply count for this gene how many mRNAs have we sequenced, and that counts 0 to 1, 2, 3, 1,000. That number is 1. So those numbers, if this is a single cell RNA sequencing data matrix, then this number is just the count for mRNA pieces corresponding to that gene within this step. So yeah, and if it's proteomics, if it's proteins, then you could be counting like proteins, different kinds of proteins. And yeah, I wanted to talk about my favorite example, which is about chromatin, but maybe next time. So yeah, summary, multi-omics just means now we can look at things at single cell level, but we can also look at different, so our P has been expanded. Our N has been expanded from 20 patients to maybe 2 million of cells. And multi-omics is about P. From each omics, you have a different kind of P. In transcriptomics, your P is RNA. And in proteomics, your P is your variables are proteins. So you can imagine we have an expansion of both N and P. So the dimension gets even higher. Yeah, let's see if I have time to, maybe I'll just briefly talk about this slide. Just to give you something more concrete. So for example, now many people use single-cell multi-omics sequencing technology to do research on COVID, because we want to understand first biomarkers, what kinds the expression levels of which genes have been affected by COVID and by disease conditions. So for example, in this study, they have patients of different kinds of COVID patients with different disease conditions, and also healthy patients, healthy people as control. And what they do is they first identify the cell types. So they will typically draw a plot 2D plot like this. So every point on a plot represents a cell, because here we have done a kind of dimension reduction. We reduce the 20,000 connector to just 2. So we use a point on a plane to represent a cell, and the colors will represent different cell types. I'm probably not going into too much detail. And in this particular paper, they want to know the difference between proportions of cells in COVID patients and in healthy people. So, and they observed that, for example, some kind of T cell is more populous in COVID patients. So lastly, I want to quickly introduce some research themes in our lab. I belong to Lacao Lab, and our head is Kim Ann. She has just been promoted to professor. So I took this picture from her website, and there was still an A there, so I covered that. So yeah, because she was just officially promoted yesterday, I think. So we look at currently we look at 2 kinds of problems. One is called data integration, and that is actually in the name of our center. Our center is called MIG, Melbourne Integrative Genomics. So we want to do integration, means it's also called fusion in machine learning. So you want to fuse or combine different types of data. It could be like different data sets, but from it could be along 2 dimensions, along n dimensions. So if we talk about so there are 2 kinds of integration. First is the graph on the left shows, top left shows n integration. So you have for example, you have a number of cells, and you can sequence RNA, you can sequence proteins, and you can sequence other things for these same cells. So if you express that you can use that, for example, using 3 matrices. And those 3 matrices have the same ends, have the same observations, but each matrix represents a different kind of variable. Could be RNA, could be protein. And then integration means how do we, for example, if we want to do a visualization, how do we use a 2D space, 2D plane to visualize a cell? So in that case, you need to do a joint dimensional reduction. So this lower dimensional space should contain information from all 3 kinds of omics. That's N integration, and you can also do a so-called P integration. So you have the same variables, but you have data from different studies, how to combine that. And in machine learning, people sometimes call this kind of problem domain adaptation or transfer learning, because it's a real problem. Because if you learn, like say if you have developed a deep learning algorithm for pictures, and suppose it's a pictures about cats and dogs. Suppose you train the algorithm from one data set. Now you want to apply it to the other data set, which also contains cats and dogs. But sometimes you could have a problem is that the resolution is different, or the way they crop they pre-process the pictures are different. So one data set is the cropping is better. So we only keep maybe cats and dogs, and maybe in the other data sets the pictures are more contains other objects like the background, etc. So they are about the same thing, and the task is the same, but you need to do a transfer because the data are slightly different. So and we have the same problem in biology. That's called P integration. And nowadays people are starting to do a sort of diagonal integration. So sometimes you don't have perfectly matching features, nor you also you don't have like perfectly matching observations or cells either. So you can imagine we don't have perfect alignment like that or perfect alignment like that. So it's called diagonal or mosaic integration. So how do we combine all those different data sets? And we can even combine data from across different technologies. We have newer technologies and we have older technologies. How do we combine those? Like from previously, we have that's too complicated. I'll just probably skip that. And the second theme is feature selection, but with confidence. So because we have a package called multi-omics, and in that package you can do P integration, you can do N integration, and you can also do variable selection. So this means when you do that joint dimension reduction, I can select, for example, 100 genes from 100 kinds of mRNAs, and I can also select 100 kinds of proteins. So I only use those RNAs and proteins to do my dimension reduction. This is important for biologists because they don't want to look at all those variables. They just want to focus on 10, 20, 50. So even if you are doing a visualization or dimension reduction, variable selection is important. So it's something like, if you know sparse PCA, it's something like that. So when you do your PCA, every PC is not the linear combination of all the variables, but only a subset of the variables. Now in high-dimensional statistics, people start to talk about feature selection with confidence. So if my lasso algorithm tells me those are the variables selected, how confident we are about the selection results? How do we know if the selection, if we have selected a false positive, or if we have omitted some important variables? So now people are trying to do inference, statistical inference on feature selection, and that's another theme in our lab. So yeah, and in the future, as I said, we're all compared to the advancement of biotechnologies, statistical methods are always slow. So we are always catching up. And already people are talking about spatial single cell, which means so that in the background you can see, so again, every point is a cell. But this is not dimension reduction. This is the real tissue. So we just take a photo of the real tissue. And so those dots are real cells under microscope. And the colors represent different cell types. So this is a tumor tissue. This is a small piece of tumor. And you can see I think the red ones are the cancer cells. Yeah, are the cancer cells. So what's different is that we have the location. We know the location of each single cell, because previously we just look at each single cell and we turn that into a matrix. But now, in addition to that matrix, we can locate the single cell the cells on the original tissue. So we have additional information, spatial information. So now we can start to do spatial statistics about single cell. And in the future, and I think under development is also, there is also spatial temporal single cell. So you can imagine if you have a embryo, you can sequence that embryo, keeping it alive, of course. I'm talking about mouse embryo, of course, not a human one. Keeping that, because now when you do single cell sequencing, you have to kill those cells as a result. But for spatial temporal, you can keep the cells alive and experiencing normal development. So at each time point, you do a sequencing, and for that sequencing, you know all the RNAs, proteins, and you also know the location of the cells. So we can even expect spatial temporal single cell data coming up in the next few years. So yeah, I think that's it. The last thing is about so just a brief summary. Single cell, we're talking about in statistics. It brings challenges to both along the n-axis and also the p-axis. And we have even like more types of data coming up. I just want to give you a very brief hand-waving introduction to this field so that you feel comfortable. Maybe in the future you attend a conference, you listen to a talk on this kind of thing. At least you feel comfortable about the words they use. Because one thing, when I first joined our lab 4 months ago, one thing I immediately noticed that if I look at the paper on Nature or Nature Communications, I can easily understand what's written in the method section, because it's statistics, mathematics, but I couldn't understand the rest. It's the rest that's important, because when a biologist look at the paper, they only look at the rest. Normally they don't look at the method section. So that's why they always put the method section at the end or even in the appendix. So yeah, if you want to collaborate with biologists and work with them more closely, then there is a language barrier. So at least but I think I've covered most of the keywords. So now it should be easier for you to attend a talk like this. The last thing is about, we were planning to continue having this maybe every month. And also there is another thing coming up next March. So we're planning to have so it's a Faculty of Science Research Summit for ECAs, for ECAs. So basically it's an internal one-day conference happening next March. But we have only just started or thinking about it. So one thing for sure, it will be free. And it will involve like all ECAs across different schools within the faculty. So it's a good opportunity to network and to collaborate. I'll keep updating you guys on my events. Yeah. So yeah, that's it. Yeah.
Cool, thanks.
Have any questions?
Yeah, with the spatial statistic.
Yeah.
How do you, I mean, or how is it done that you sort of use that information and combine it with the sort of that more your big data sets of cells and genes? Well, it's not clear to me as to how
because
you fit those together.
I can that's true, because the truth is right now people there aren't many. So okay, for example, how do we get a picture like this? They didn't do any spatial statistics. They just treat it as normal single cell, and they do clustering and they annotate those cells. So basically they identify the cell types using conventional methods, and then they color the picture according to that result. So while they were doing statistics, they were not using any spatial information. And currently that's the case for most of the studies in spatial single cell. But if you want to use that, and we know how to do that in statistics and in machine learning. For example, if you want to do clustering, then at least you can impose some restriction. You say, okay, so if two cells are spatially close, then we tend to think they might belonging to the same cluster or the same cell type. Yeah, you can impose that as a hard restriction or a soft restriction. You can introduce some distance or or some because in spatial statistics they have all kinds of spatial random process. For example, you can talk about spatial Markov process, which means the distribution of one cell, for example, only depend on cells around it. So things like that.
So it would be about identifying the type of cell.
Yeah.
Using a spatial model.
Yeah, that's
okay.
Yeah, spatial clustering. Yeah. So and if you use that, then as an immediate visual result, you will get a picture slightly cleaner than this. You will see like a better spatial. Of course, this one is already good, but because this is on a this is from they definitely picked the best example to put on their website. But yeah, I heard from people that this kind of data is very messy. So normally you couldn't get a nice picture like this. But if we do real spatial statistics, then maybe we can get something better. Yeah.
Cool.
Yeah. And I didn't ask you, Jason, what kind of applied math?
So I'm in fluid mechanics. I do a bunch of different continuum mechanics stuff, mostly in physical problems. I do a tiny little bit of work with biologists. We do some measurements on measuring the stiffness of cells. But yeah, mostly I'm looking at things like measuring and quantifying nanoparticles. Yeah, a bunch of different stuff in kind of the physical applied.
Yeah. So do you deal with like in terms of mathematical tools? Do you use sort of
pretty much all differential equations? Yeah. Most of my skills are in kind of analytical methods for solving differential equations.
Oh.
Yeah, I do a lot of asymptotics, singularity methods, transforms.
Yeah, I think that's because I have I had some experience working on traffic problems, because in traffic problems they also have applied mathematicians working on they really model individual cars, etc. But also statisticians tend to look at the big picture, just statistics.
Yeah, and there's a lot of people who use continuum models in traffic modeling as well, which is really interesting. Yeah, because I taught applied mathematical modeling for a couple of years. And there's a section in that on traffic modeling, and it's some basic PDEs stuff. They use method of characteristics to solve for things like looking at shocks and fans when you're doing traffic modeling.
Oh yeah. I actually took a subject on that. A few years ago, we had an MC summer school, and one guy from New Zealand, he worked on wave functions, and he used that to do traffic.
Remember who it was?
Not really. Yeah, but I suspect you might know that guy's name, because he's like one of the few guys working on particular type of.
I did.
I've seen the colloquium on that.
Really?
Modeling traffic.
Yeah.
Yeah. Well, maybe that's a common thing. I'm not too.
Yeah. Was the speaker a male speaker from?
Yes, I believe so. It was quite a few years back, so I don't remember it too well. So what were you going to say, Jesse?
I did my undergraduate in New Zealand.
Oh.
I did it in engineering science, and there was kind of like a subspecialization of optimization and traffic modeling and stuff. So yeah, it's entirely possible I had this person as a lecturer, but
Yeah.
that was quite a while ago.
Yeah. The most shocking thing for me was I learned that they talk about a kind of phenomenon called ghost jam. Ghost jam that can be explained.
I think it was the same.
Yeah.
Sorry, go on.
Yeah. No, I was I mean, it's very interesting. Like you can, like if nothing happens on the highway, you can still get traffic jams. It's because of that. Yeah, propagation.
Yeah, it's really fun when you're in traffic trying to figure out the wave that or which part of the wave you're currently in. And when you're like driving along the freeway, sometimes everyone will just slow down and then you speed up again. And it's just because you're in a different part of the wave.
Yeah. And I think the suggestion I received from that lecturer was to keep a long distance. Because because you can. That's also what experienced drivers do. They keep a long distance from.
Well, basically, you can either act